Hi guys, it's Mrs. Vance again. This recording is the first part of notes for Unit 7, which is about the structure of DNA and how DNA copies itself, and also the cell cycle, which is how cells go about copying themselves. And so we're going to learn about DNA first and about how it copies itself, and then we'll learn a little bit more about how the cell makes copies of itself as well. So the first part is about the structure of DNA. Okay, and I like this cartoon because it kind of encapsulates what we think about DNA most of the time. DNA molecules, basically a list of a to-do list for everybody else in the cell. And so basically that's what it is. It has the instructions for making all the proteins, and it's considered the control center of the cell, but it's really not the control center as much as it is the main recipe book for everything that needs to be done by the cell. So again, we've got some vocabulary words that we need to focus on, and we'll talk about some of these. We'll talk about these as we go through this part of the unit. But some of these you've learned before. We learned about nucleotides back in our biomolecules unit, and we talked a little bit about DNA structure then, and about nucleotides in specific, and then we'll talk a little bit more about how it fits together, right, and how it fits together a certain way, and how it makes copies. And so that's what all this is about. So DNA and RNA are what we call polynucleotides. They're polymers of nucleotides. Okay, remember that DNA and RNA, nucleic acids, are made of nucleotides, and nucleotides are composed of a nitrogen base, a 5-carbon sugar, and a phosphate group. The picture here on the slide is actually a DNA nucleotide for the nitrogen base thymine. And so you can see over here you've got your phosphate group. Here's our sugar, our deoxyribose, okay? And then here is our thymine molecule. This is our nitrogen base. And there are 4 different nitrogen bases that we'll talk about one at a time and talk about what the difference is. But this is nitrogen base. This happens to be the T, the thymine, but it can be either adenine or guanine or cytosine, as well as thymine, okay? And then the nucleotides are joined together by this sugar phosphate backbone. Basically, this phosphate will attach to this part of the next sugar in the chain, and it just goes on up and repeats as you move up from the bottom to the top. So we'll talk about how that works in just a minute. Each type of nucleotide has a different nitrogen-containing base. You can see here the 4 different bases. We have thymine and cytosine, both of which belong to a group of nitrogen bases called pyrimidines. You can see that they have a single ring structure with lots and lots of nitrogen in it. And then the other 2, adenine and guanine, look a little bit different. They have a double ring structure, and they're what we call purines. It is important ones are which. That's important. And so adenine and guanine, ags, are purines. And so ags are pure. If you can remember ags are pure, that helps you know that adenine and guanine are the purines. And then thymine and cytosine are in the other category, which is the pyrimidines. And that one I don't really have a handy mnemonic device for. Just remember that they're the other ones that are different from the purines. So each one of these has a different nitrogen-containing base, but the basic part of the nucleotide is all the same in all of them. So here we see how they join together. Basically, you see that they can connect together through that phosphate, sugar phosphate, sugar phosphate, sugar phosphate there. That's what we call our sugar phosphate backbone, okay? And then the nitrogen bases are attached to the sugar molecules, the deoxyribase in the case of DNA, okay? There are a couple of different ways this is illustrated in books, okay? So here you have a double helix model that shows you the paired bases in the sugar phosphate backbone. This is a simple model showing you basically that you've got your sugar phosphate backbone here, and then you've got the nitrogen bases attached. This one shows you more about what the structural molecules would look like. And then this one shows you specifically that DNA nucleotide for thymine that we looked at just a minute ago. So we've got our phosphates, and there's our sugar. You can see this is deoxyribose because there's not the only oxygen. There's an oxygen here and one up here, but that's the only oxygen. The D part means remove oxygen, so there's one less oxygen here. And then our nitrogen base attached to the number 1 carbon. And sugars, if you think back to when we talked about biomolecules way back close to the beginning of school, we said that you can number the carbons if you start with the oxygen and start going clockwise around. The first carbon you come to is number 1, 2, 3, 4, and 5. So there's our 5-carbon sugar, and that it's important to understand the 3 and the numbers of the sugars, because that's important to the way DNA makes copies and how it's organized. So RNA is also a polynucleotide, but not polynucleotide, but instead of deoxyribose, it has ribose sugar. And if you think back to the one we just looked at in the previous slide, this one here, notice that on this number 2 carbon, there's only a hydrogen attached, but on the ribonucleic acid nucleotide, or the ribonucleotide, there's an OH there instead of just an H. So this one has one more oxygen than the deoxyribose does. So this is ribose sugar. Notice we've still got our phosphate group attached to the number 5 carbon and our base attached to the number 1 carbon here. Another difference between RNA and DNA is that RNA has the base uracil instead of thymine. It's shaped very similarly to thymine. I'm going to switch back and show you that one. Here's thymine. Okay, and you see that it's very similar in its structure to what we see in the uracil. Very, very similar. But thymine doesn't fit with the ribose sugar the right way, but uracil does, and it fits together with its paired base adenine as easily as thymine does. Here's a picture of an RNA polynucleotide, just showing you one of the various arrangements. Notice that generally the RNA is going to be a single strand rather than that double helix. But there are lots of different shapes that RNA can take depending on the function of that particular molecule. DNA is a double-stranded helix, okay? And this was discovered back in the early 1950s. In 1953, James Watson and Francis Crick figured out the secondary structure of DNA. They knew about the nucleotides, they just didn't know how they were organized. And so they used a lot of different data that other people had collected. They used the data of DNA from the work of Rosalind Franklin and Maurice Wilkins, who were working on X-ray crystallography of the DNA molecule. And another scientist, Erwin Chargaff, had figured out that in DNA, the amount of adenine was always pretty close to equal to the amount of thymine, and the amount of guanine was equal to that of cytosine. And so Watson and Crick kind of put these two ideas together, and knowing that there's a certain way that it was organized. Here's the picture of the X-ray, and knowing that this shape here, this round with this crisscross in the middle, indicated that it was a double helix. This is Rosalind Franklin who figured that out, okay? And Watson and Crick managed to get a copy of her picture, and that led them to believe that it was a double helix. And then using Chargaff's information about the comparative amounts of the bases, they figured out that it was a polynucleotide strand that wrapped up or twisted up into a double helix with the sugar phosphate backbone on the outside and the nitrogen bases perpendicular to the backbone in this interior. Because the bases are arranged the way they are, that gives the helix a uniform shape. A pairs with T. A is adenine, which is a purine, and T is thymine, which is a pyrimidine, and they form 2 hydrogen bonds connecting them together. And then guanine, another purine, pairs with cytosine, another pyrimidine, and they form 3 hydrogen bonds between them. And because you have a large purine with a small pyrimidine in each pair, that gives them a uniform diameter to the double helix. So here's Watson and Crick. This is James Watson. He was an American scientist. He was about 26 years old at the time. And then here's Francis Crick. And this is showing the model that they built out of wire and ring stands and cardboard pieces that they put together to kind of show how the molecule fit together. They won the Nobel Prize in 1956, I'm sorry. Their idea about the double helix was that basically you have a ladder. The sugar phosphate backbone is forming the sides of the ladder, and then the paired nitrogen bases are forming the rungs or the steps in the ladder. And if you take that ladder then and twist it, that gives you that double helix shape. So here we have our ribbon model showing our double helix here. Again, we have the sugar phosphate backbone shown with the ribbons on the outside, and then our paired bases showing there. This is a computer model showing basically, it's called a space filling model, that shows you basically what it looks like with the orbitals of the electrons around the atoms. And then this is a partial chemical structure that was more familiar to us, like our usual structural formulas that we look at. Notice that C and G, again, have 3 hydrogen bonds between them. T and A have 2 hydrogen bonds between them. And if you change the order of the bases of one side, the other side is also going to change. And then we call these complementary base pairs, complementary with an E, because they complete each other. And notice that gives us a uniform diameter here. And they're all connected up to the number 1 carbon of the ribose, or of deoxyribose rather, and they're connected through the phosphates, through the number 3 and the number 5 carbons here on that side. Notice also when you look at the picture that on the right-hand side, the sugars are upside down. That's because the molecules actually don't fit together right until you turn one side upside down. This is called being anti-parallel. Instead of parallel, going the same direction, one goes in one direction and the other side goes the other direction. And that's a really important thing to understand about DNA structure. So here we have, basically, once again, we have our ribonucleotide here with uracil, and our deoxyribonucleotide here with thymine, showing that they are very similar in structure, except for the difference of the sugar and a couple of differences in the functional groups that are attached to the ring structure there.